Okay, guys, let's get started. Just quickly check if it's the correct link or not. Okay, yeah. Okay, welcome to week 12, the final week of the course, Next Generation Sequencing Technologies and Data Analysis and Applications. So, in this week, we'll go through epigenomics and different uh, technologies used to detect epigenetic modifications in the genome. And first of all, we'll study about why we need epigenetic modification, why we need to study epigenetic modifications, why are they important, and after that, we'll see some next generation techniques used to identify these different patterns of epigenetic modifications across the genome and implications in disease and other conditions. So, yeah, let's get started. So, if we talk about gene expression regulation, the central dogma of biology actually tells us so from we get DNA, the DNA replicates itself, then from DNA we get RNA, from RNA we get protein, and this whole concept of flowing of information from DNA to RNA to protein is very tightly regulated through different kind of mechanisms. So on the gene level, if a gene is transcribed into an mRNA or not, that control is on the hand of is given the attribute of gene expression, uh, control of gene expression. Now, gene expression can be used, can be controlled in various manners. Now, uh, like say for example, we have the same set of we have the same DNA in the liver cell as we have the same DNA in a lung or a nerve cell, but the physical characteristics and also the mechanistic differences between these type of cells are attributed to the fact that we have the same, we do not have the same expression of genes and proteins across these different types of cells. Now, why is that the case? There are different levels of regulation of genes. So, if like if in the liver cell a particular gene is getting expressed, say for example, uh, some bile secreting enzyme is getting expressed in the liver. So in the because liver has to secrete bile. So in liver, that particular gene or enzyme will be expressed. But again, that same gene is also present in other cells of the body. So for example, the nerve cell or the lung cell or stomach cell won't be expressing that particular enzyme or particular gene to that level that is expressed in the liver. Now, this control of expression of gene can be at different levels. The major part is in the transcription level, so where basically a transcription factor will come and bind to the promoter, and then the sequence, then the mRNA formation will start. Now, this control, this coming of the transcription factor and binding, and then starting of the transcription of the gene is highly regulated. So it is highly regulated when a transcription factor should come and bind, and at what position it should bind to give a good mRNA. Along with transcription factor, there are other parts of the DNA which are involved in this gene regulation. There can be upstream elements like enhancers or repressors. There can be other types of elements that can come and like although they are quite upstream to the gene that is actually to be transcribed, but those uh, elements upstream or downstream can affect the transcription of this gene because in a 3D structure, these particular elements they come and very close to this particular gene sequence. So physically they are brought together in a way that it can influence the expression of the gene, but yeah, so there can be many elements to it. So one such element is epigenetic regulation. So how does epigenetic regulation works? So there are different enzymes inside the cell. So first let's talk about DNA assembly. So in general, so there's something called the histone protein in eukaryotes. So the DNA is not naked, and it is well packaged around histone proteins. So these histone proteins are generally octameric proteins having 8 subunits, and these proteins are positively charged. On the other hand, your DNA, like courtesy of the phosphate group present in the DNA, it's negatively charged. So it becomes easy for the DNA to get wrapped around this particular histone proteins, and what they form are like what are known as beads on string structures. So basically, you have a stretch of DNA, then you have the histone protein and DNA wrapped on the histone protein, then again some free DNA, again histone protein and DNA wrapped on that. So this is how the beads on string structure is getting generated. But these histone proteins are very important in regulating gene expression because uh, there will be this packaging is not uniform throughout the whole DNA sequence. And so there will be places where DNA is packaged more compactly, more uh, in a more compact manner, and there will be places in the DNA where it is not, the packaging is not so compact. So now this packaging controls the accessibility of the DNA to different kinds of transcription factors, different kinds of proteins that will come and help transcribe that particular gene. So the accessibility of the DNA is controlled by this particular packaging material, ultimately histones, and the histones on the other hand are also are regulated, like how they should bind and how, at what point it should not bind. So this is also regulated by certain other enzymes, primarily histone methyl transferases, which are known as HMTs, and there's another set of enzymes known as HATs, which are histone uh, acetyl transferases. So there are these set of enzymes along with the help of the histone proteins, which kind of control the accessibility of the DNA and in turn control the regulation of the expression of that particular stretch of the DNA along which it is wrapped around the histone. So as I was saying, you have the central dogma of biology, so your DNA transcribing to RNA, then to protein, and this gene expression levels, that is uh, quantity of RNA, mRNA forming from that particular region, is basically the gene expression, what we define as a gene expression, and the amount of gene expression and the amount of expression of particular genes in particular cells is what gives the cells particular characteristics. Uh, Beta nerve cell, beta skin cell, or even in disease conditions, the gene expression will vary. Now, as I was saying, the DNA methylation is one of the epigenetic ways to control gene expression on an accessibility front. So, DNA methylation is basically adding up of methyl groups on certain positions in the DNA where like on the basis of which the accessibility of the DNA is controlled. Now, the DNA methylation patterns are also like it's not a very consistent pattern across different individuals or different types of conditions or different types of cells. So, depending on the expression of the gene that is required, there will be various kinds of signatures of the DNA methylation inside the genome. So, this is one such example where basically these graphs are showing methylation patterns inside the DNA. But this is an example where it is showing the differences in DNA methylation pattern across a 3-year-old twin and 50-year-old twin. So although they are twins, I can see they're distinctly different in terms of the methylation patterns in the DNA, and ultimately which is giving rise to different expression, different gene expression and distinct characteristics. Okay. So as I was saying about chromatin organization, like you have the whole set of the DNA inside the chromosome, and in the condensed form, it is what is known as chromatin, and in along the chromatin, you have packed fiber of packed nucleosomes. So basically, your histone octamers are present on which the DNA is wrapped. So it's around 147 or 150 base pairs of DNA separated by linker DNA. So you have this like around 2.5 wraps of DNA around a single histone protein, and like two of such histone proteins are separated by some part of the linker DNA. So and this particular formation is so the DNA wrapped around this particular. Yeah. So sorry. Yeah. Uh, so the DNA wrapped around this particular histone proteins, these structures, bead-like structures, are called as nucleosomes, and this whole organization of chromatin with the histone octamers are known as this whole organization is called a beads on string formation. So you have different levels of chromosomal organization. You have at nucleosomal scale, which is basically one PB to 10 KB, around that in that particular range. If you go down further and you look at more microscopic level, so you have this topologically associated domain. So basically, although in terms of the sequence, a particular sequence will is away, is say for example 500 MB or 1 MB, sorry, 500 KB or 1 MB upstream or downstream of that particular area, but in terms when it is packed around this chromatin structure and forms a 3D structure, it actually physically they are brought together. So as I was saying, like enhancers, elements like enhancers and repressors, they are actually very upstream of the gene that it is supposedly regulating. But what it forms is from like a hairpin like structure, so that even if it is up, it is very upstream of that, so it folds and brings that particular element closer to the gene that it is trying to control. So you have these topologically associated domains, which are the pieces of DNA, although they are far apart if, in terms of the sequence, but in the 3D structure, they are brought together due to the organizational structure of the chromatin. And then you have this inter tad dynamics, where you have this AB chromosomal compartments. So this is an chromosomal compartment, and this is a B chromosomal compartment. These are known as chromatin loops. So, and these kind of compartments are joined by linker DNA. So inside these are nucleosomes, and all these nucleosomes and this topologically associated domains and these compartments, they come together and condense and form the nucleus. So at the nuclear scale, it is like much at a macroscopic level. So this is basically at the level which the epigenetic modifications happen. So we start with a like a double helix structure, then we wrap this double helix around a particular protein, positively charged protein. Then we formed different loops of this particular protein plus DNA complex, and we form small structures called as nucleosomes, and which are compartmentalized. A few nucleosomes are compartmentalized in one place, forming different chromosomal compartments, and which are again condensed together to form chromosomal territories and ultimately the nucleus. Okay. So, as I was talking about, there can be different types of patterns of this particular methylation. Um, uh, so there can be particular methylation patterns inside the DNA, depending on the accessibility of the which regulates basically the accessibility of the DNA for transcription. So these are some of uh, the symbols that is used. So for example, if it is showing an H3K4Me3, which means is at the histone 3, at the lysine residue, fourth lysine residue, there is a trimethylation. So these are what the symbols mean, so the representation means. So if it's a third histone lysine residue three times trimethyl. So these are active promoters. You have other signatures like H3K4Me1 and H3K4 K27AC. So this is an acetylation. This is not a methylation. So these are signatures of active enhancers, and you can have signatures of promoters and repressors as well. So the H3K27Me3 is a expression repression example at promoters, and H3K9Me3 is a signature of heterochromatin, which is basically inactive and condensed chromatin. So there are two types of chromatin possible, along with that structure that we have, the beads on string structure. So one is a heterochromatin where it is highly condensed and they are highly packed together with the histone modifications, and they're clubbed together in a very tight space. So these are called as, and these are, if you see through a microscope, these will be like very dark areas inside the in the genome, and these are what are known as the inactive and the condensed chromatin areas, which basically you do not have enough access to the DNA for the transcription factor or the protein to go and bind and affect a particular transcription or whatever it is doing. So you have places in the DNA where it is very low accessibility of the DNA is present. So those are known as heterochromatin, and you have places in the DNA where the accessibility is very high, which have like basically which are known as euchromatin. Now, this is a chemical representation of how DNA methylation and demethylation occurs. So, the addition of a methyl group at the fifth carbon position of the cytosine base. So, basically, this is your cytosine base, and at the fifth position, there is a methylation that is occurring. So, these are all the enzymes that are used for methylation. So DNMT are methyl transferase enzymes. You can have DNMT enzymes. You can have acetyl transferase enzymes as well, HATs. So depending on if you want to open up the chromatin or if you want to close the chromatin, depending on that, these enzymes are used. So this process is mediated by DNA methyltransferase enzyme family. DNA methylation is mediated by 10-11 translocation, which is the TET enzyme family. And active demethylation is a sequential process in which 5-methyl cytosine is converted to 5-hydroxymethyl cytosine, which is 5-HMC, which is converted to 5-formyl cytosine, which is 5-FC. So these are different, so you will come across these in the later stages of this lecture as well. So these are what is meaning, yeah, in short form is basically 5-MC. HMC means is like the conversion of 5-methyl cytosine to 5-hydroxymethyl cytosine, which is basically a formation of a hydroxy group uh, along with the methyl. So this is how the formation occurs. Now this process readies the site for thymine DNA glycosylase, sorry, thymine methyl glycosylase, which is known as TDG, to remove both of these 5-formyl cytosine and 5-carboxy cytosine. So these processes readies the site for this particular enzyme to remove these intermediate products. Uh, so this is the whole reaction that is happening. So you have these DNMT enzymes which bring methyl groups here. So you have this formation of this 5-NC DNA, which is further enzymatically converted to 5-HMC by TET enzymes, and then you have formation of these intermediate products, which are 5-CAC and 5-FC, and ultimately this is the demethylation process, and where this TDG enzyme comes and removes these, and you get the original cytosine back using base excision repair. So this is a complicated process. We'll not go into the details of this process, but just to give an overview why we need to study it, because depending on the methylation pattern of the DNA, you can predict which part of the DNA is active and which part is not. So if you can predict that, you can predict the genes that are present in that particular portion of the DNA is might be active or inactive. So yeah. Uh, so There can be many ways to study DNA methylation patterns. So next generation techniques are obviously there, but there can be very simpler techniques as well. So if you say, for example, we can use DNA methylation analysis, uh, and we can use restriction enzyme based models. So these kind of restriction enzyme based techniques allow the study of the CPG islands. So these are like small areas of the genomes, these islands of the genome which are cytosine rich. So this CPG island methylation of individual genes and disease or pathway focused gene panels without bisulfite modification. So bisulfite modification and screening is a widely used method for studying epigenomic modifications, but those are a little bit more complicated, but if you can obviously study these using restriction enzymes as well. So it relies on differential cleavage of target sequences by two restriction enzymes, endonucleases whose activity required either the presence or absence of methylated cytosine in the respective recognition sequence. So depending on that, if it recognizes a methylated cytosine near or uh, just uh, just beside the its restriction site, it'll either activate or it will not activate depending on the type of restriction enzyme you're using. The relative amount of DNA remaining after each enzyme digest is quantified by real-time PCR, delivering reliable calculation of methylation status of individual genes and methylation profile of across a gene panel. So basically, you use specialized restriction endonuclease enzymes which identify this particular methylation patterns in the genome, and depending on the methylation pattern, they kind of will act or not act. So if it acts only on the places of the methylation pattern, so it will give you stretches of the DNA where there is a good amount of methylation and stretches of the, and you can check those particular, and you can basically amplify that particular stretch of the DNA using PCR and check where from it is coming. So it can give you a decent idea about what is the methylation status of individual genes and what is the methylation profile across a gene panel. So the next kind of techniques that we use is basically bisulfite based. So there is a very useful technique for this. So if there's a uh, methylated. So basically, this is bisulfite based indirect techniques. These are bisulfite free indirect techniques, and these are bisulfite free direct techniques. So bisulfite based and indirect methodologies encompass bisulfite sequencing for both 5-MC and 5-HMC, uh, oxidative um, oxidative bisulfite sequencing for 5-MC, and TET assisted bisulfite sequencing, which is the TAB-seq for 5-HMC. So these are different kind of signatures that is identified by this particular technology. So uh, this 5-MC and 5-HMC can be identified by both by sulfide sequencing, which is BS6. Oxidative bisulfide sequencing can identify 5-MC, and TAB-seq can identify 5-HMC. Bisulfite-free yet indirect methods include enzymatic methyl-seq, which is EM-seq, for both 5-MC and 5-HMC. And there are other methods, coupled epigenetic sequencing for 5-HMC, which is known as ASeq, so apo-bake coupled epigenetic sequencing. Alternatively, bisulfite-free and direct strategies entail chemical-assisted C to T conversion of 5-HMC. So basically, where there is a where there is a 5-methyl, where base is cytosine base is 5-methylated, so that can be it can be converted from a C to T, depending on the type of technique you're using. So where there is methylation, you'll either have a conversion of that particular base to a different base, or that particular region of the genome can be explicitly identified if you just use restriction and enzymes and just cut off the areas next to it. Okay, so just a second, guys, I'll be back.